talking about what recombinant DNA technology is first. It's basically utilizing our understanding of how DNA transcription and translation work to our advantage to manipulate organisms to make products for us or to change the product so that it works better for us or something like that. So we can fix mutations. That way we could even take somebody's DNA that is messed up right now in this room, and there are potential treatments that can change your DNA in your cells as they already exist right now and make it so that we could change that. So that's the idea of how recombinant DNA technology could work for us, okay? We can change that based on our knowledge of how DNA and transcription and translation all work, all right? So one thing that we need to understand about DNA is that if you take a double-stranded molecule like this and heat it, those hydrogen bonds that were holding those G's and C's together, so G binds with C or A binds with T, that this bond here is just a hydrogen bond. And that is just based on attraction. Okay. So it's easy if you heat something up to weaken those bonds. They're weak bonds. So adding heat to DNA will allow us to separate it. But if you inversely, if you cool it down, all those A's and T's and G's and C's are going to want to bind back with each other. And so they'll just come back together exactly how they normally would be, right? So this is going to be a driving concept on a lot of the stuff that's going to be talked about in this chapter. Another thing that we need to understand, this is the only time, well, the second time, we had to know how to calculate our power on our microscope. Your total magnification of your microscope, which was essentially your eyepiece times your objective lens. And our eyepieces are 10. So I'm asking you guys to multiply by 10 with that, okay? So there was that one. This is the second and last of all the math that I will ask you to know in this class, okay? And this does require that you understand things like the metric system and how they are asking you to understand that stuff. So we have base pairs. Everything in your DNA, if your DNA read GATTACA, which hint, 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 the whole reason that movie is named that is because G's, T's, C's, and A's for Gattaca, that your other sequence would be G, C, T, A, T, G, T. Okay, that's what your two sides of your DNA would look like. And this would be your 5, and this would be your 3. Okay. And that's what your double strand looks like, and that everything is linked together by those hydrogen bonds and just to reprise kind of our gist of what we learned about DNA and how it functions. When we're counting how big your DNA strand is or how big a sample of DNA is, we count it by base pairs. This is one base pair. So this is 1, this is 2, this is 3, and so on and so forth. That's a base pair. So if your coding strand read Gattaca, we would know what your other strand said because we understand how G's and C's and A's and T's bind together. And that we would say that this is 1, 2, 3, 4, 5, 6, 7 base pairs long. And that's how we basically talk about how big someone's genome is or how big a fragment is. And that this stuff is going to be useful in basically comparing DNA pieces, which is another thing we're going to talk about today. All right, so we will measure things by how big their genome is. For example, an average gene, just a single gene, not a genome, just one gene that codes for one protein. In E. coli, the average gene is 1,300 base pairs, or 1.3 kilobases, or kilobase pairs. Anyway, you guys need to understand that kilo means multiplied by 1,000. Okay? So we have base pairs. 1 is a base pair, 1,000 equals a kilobase pair. And then we even have up to 1 million, which that one is a mega base pair, MVP. All right. So that's basically what this is talking about, is comparing some things. So the entirety of, if we look at all of the genome of Epstein-Barr virus, which is what causes mono, okay, mononucleosis, that Epstein-Barr virus's genome consists of 172 kilobase pairs. Okay. The entire genome of E. coli is 4 million. I'm not going to test you on these numbers, right? But understanding when if I write this, if I said if E. coli's genome is 4,700,000 base pairs, that is the same thing as 4,700 kilobases, or 4.7 mega. Okay, those conversions. I'm only going to ask you about base pair, kilobase pair, or mega. All right, know what the difference is. You see down there what it says about human genome? 6 billion base pairs. Okay, pretty crazy. All right, CRISPR. You may have heard about this in genetic engineering and updates in science and everything like this. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. I'm not going to test you on that, but I do need to know that CRISPR and Cas9 and what those things mean and how they work. Okay, so in nature, this enzyme Cas9 in this whole setup of CRISPR recognizes and cuts out foreign DNA. All right, so normally in bacteria, in science, in nature, this system of CRISPR and Cas9 work to eliminate foreign DNA. We can manipulate how Cas9 works on certain sequences with genetic engineering to target potentially any genetic sequence anywhere we want. Okay, and this is useful because it means that then I could use CRISPR-Cas9's system to go into your genome and repair in your actual genome, in your live living cells, that target sequence of DNA. I have to know the sequence and I have to be able to manipulate CRISPR and I have to be able to get into your cells and actually be able to make these manipulations. The realistic aspect behind the usefulness of CRISPR has not been realized yet. Nobody has been able to successfully utilize CRISPR to cause genetic changes in living organisms yet. We can do it at a single cell level. We can do it at like bacteria and things like that. We can control that, but being able to do it at the macro level and multicellular organisms hasn't been realized yet. But there's plenty of research being done on it. And you can imagine the effect that this could have on human society if you're having let's say you're having an issue where you can't make insulin. You have a genetic defect where you literally aren't making effective insulin and something like that. We can go into giving you gene therapy treatment as a living human being that's already been born and already has this defect and treat you with this system of CRISPR and Cas9 to repair that. Yeah. Right? It also will allow us to change your eye color, your hair color, your height, things like that. That's called eugenics if you don't know. And so this is a slippery slope argument of where this becomes ethical. And if you haven't seen the movie Gattaca, this is what this is referring to, okay? In Gattaca, you have a parents that had 2 kids. One of the kids was naturally born, the other one was genetically engineered. And that in that society, if you are genetically engineered and you meet these qualifications of being genetically engineered, you can do these jobs or these jobs or these jobs. You're basically engineered to do those jobs. And that people who are born naturally cannot possibly be able to meet the requirements to do the jobs that you're genetically engineered for. So this man says, I want to be an astronaut. I was told that because I was naturally born and I have this heart defect because of my natural birth, that I can't be an astronaut. So he works his ass off to fake it. And he basically steals somebody else's genetic identity and steals, like, they work with him to provide his Jude Law. And I just highly recommend that you look at that. It gives that sense of where the problems might actually lie in looking at it that way. Gattaca. So Gattaca. Yes, it's an old movie. It's from like the 90s or something. It's got Uma Thurman. Right, exactly. So this is what this discussion is. Is there an like the ethics of it, right? And so he fools everybody into thinking he's been genetically engineered. And he trains and trains and trains. It proves that he gets to go into space and he gets to be an astronaut. But at the same time, there was a reason why he shouldn't have been allowed to be an astronaut, because he has a heart defect. And now if he dies while he's being an astronaut out in space, the people that are out there in space with him now have to deal with the fact that they have somebody die from their heart defect out in space. There's a reason why that rule was in place, right? But the ethics behind everything leading into that. That's the problem with genetic engineering on that level. And at which point are you going to draw the line of, well, this person has this genetic defect that's going to make them less capable than another person, but they would be able to overcome it due to disability aid and things like that. Where does it become unethical to be changing that genetic information and questionable at best? So if you aren't thinking about that stuff, this chapter is definitely going to be leading to that. I have a video on CRISPR, but I'm going to skip it. If you guys want to go back and view it yourselves, I just don't want to keep us here too long. Okay. All right, moving on to restriction endonucleases. These are enzymes, okay? So another enzyme that's going to cut DNA at a specific sequence. But these guys can only recognize, we can't engineer these guys to cut any sequence. These guys are made, pre-made to only cut at certain sequences. They recognize sequences of 4 to 10 base pairs. So they might be able to see something like if you had GAA, TTC, okay? This might be a sequence that a restriction enzyme and there's stuff connected to it, whatever, there's the rest of the DNA. And then on the other side of this, I would like to point out that this sequence would match up as CTTAAG. This is important on the other side of that ladder because we're going to talk about what restriction enzymes are and how they do their recognition and cut and everything like that, okay? So if this is your whole, sequence, they would recognize something like this. So I want to point out GAA-TTC. You see how these guys are kind of inverted from one another? So it's like half of it is the inverse of the half of it that's attached to. And that if you read this one, GAA-TTC, what does this one up here read? The same thing. Oh, let me move this. That's fine. Yeah, go down there. GAA-TTC. So this is what we call in DNA a palindrome. It reads the same forwards as it does backwards, like the word race car. Or yes. Or taco cat. Yes, that works too. But so it's a palindrome. It reads the same forwards as it does backwards, but forwards on one strand versus backwards on the other strand for DNA. Okay. And that the fact that sequences like this exist, these enzymes will be able to recognize that and then cut them at those palindromic sequences. So it's a sequence of 4 to 10 base pairs that's palindromic. All right, it's normal, just like with CRISPR, it allows bacteria to get rid of foreign DNA. So if it sees a sequence like this, it'll just cut it and that keeps that DNA from being able to function. So we can take up this foreign DNA and maybe we got a plasmid from the guy next to us, but it has a bunch of crap on it that we don't want. Maybe these restriction enzymes will cut at these palindromes and get rid of the DNA that we don't want. Okay, so that's basically how they will work. So they work in palindromes. It's identical, read in one direction versus the other direction. So if we, again, I'm going to put it back up here so we can visualize it. GAA-TTC, and then GAA-TTC, right? And this is our strand here. Now these guys will cut at these palindromic sequences, but they don't just cut like this. Sometimes they will cut like that, and that's called a blunt cut if we do that. So this is blunt. But most of the time, the thing that we use them for and the thing that we find them to be useful for in genetic engineering is when they don't cut like that. They cut instead like this. All right. And so then your products would look something along the lines of, aren't you guys glad that I actually have the pen today and I'm not trying to write with it for the mouse? Can you imagine? GAATT. And then the other side would be AAG. And then, come on. TT. That's what I'm missing. Why does this not look right? All right. And so you would have these things cut apart and look like that. So you have these overhanging bits of sequence here that we call sticky ends. So it cuts them there and they're not attached to each other anymore. And that is what we call a sticky end. They're symmetrical and they're staggered to create those overhanging bits of like single strand kind of nucleotides. Okay. All right, so this is important because now you can engineer a human gene and put those little tags at the end of it and just pop it into your DNA sequence for your bacteria. So often we can make plasmids that this is a whole plasmid, a whole ass plasmid, y'all, that we could cut with a restriction enzyme to give these overhanging sticky ends. And we've cut our DNA from our whatever organism, maybe it's human, I don't know. And it has sticky ends, and now they will insert that guy into your plasmid. And there's ways that we will learn about in the lab that will allow you to make sure that the plasmid that you're putting back into your bacteria has that gene inserted into it and didn't just like reclose back in on itself. Because that's another concern people will have. Like, wouldn't that plasmid just stick back onto itself? If you give enough excess of the thing you're inserting in from your other organism, the odds of you inserting it is going to be much higher than it closing back in on itself. And also, on top of that, we have ways to test for that organism to make sure that it actually inserted that in there. We can make sure it inserted it into the plasmid, and we can make sure that we're only growing up bacteria that got the plasmid in them, because that's another problem too. We're going to learn about that in our transformation lab. That's like lab number 10. Okay. So that's the idea of how those restriction enzymes work on creating sticky ends. And that's why they're useful. We're starting to see that, at least the gist of it. Okay. So you can just have these sequences randomly in your DNA. So if I cut with an enzyme, and an example of one of those they showed us over here, and I won't test you on it, but I'm just going to use it for sake of explaining it. Might be one called EcoR1. EcoR1 literally is named because it is from E. coli, so ECO, restriction enzyme 1. That's what it's named for, okay? So EcoR1 is a restriction enzyme that cuts this way. It makes that GATTC that we were just talking about and cuts it with that overlap, just like what we were just talking about, okay? So that's EcoR1. And that EcoR1, you might have randomly, just randomly in your DNA sequence, GAATTC. And that if I cut your genome with GAATTC cutting enzyme EcoR1, that you'll create these fragments in your DNA as a result of that. All right. So if I cut yours, you might have 5 fragments as a result, just from random sequence in your DNA. But if I cut mine, I might have 10 fragments because I just have different DNA than you, and I might have more cut points for it because of my sequences. So now are you starting to see that if, let's say, somebody came up here and murdered Professor Yost?
No.
Yeah. And there's blood, and it's not all mine. And so we're able to discover, oh, there's blood up here. There was a scuffle. Professor Yost is dead. Who murdered Professor Yost? So I can get blood from all of you guys, and I don't have to sequence your whole genome. I can literally just cut it with these enzymes and see what bands show up, what fragments of your DNA, and then basically look at the sizes of those fragments. And the likelihood of somebody having the same pattern as another person is extremely low, even if you're related. It's extremely low because we have, what did we just say, 3 billion base pairs? Yeah. Identical twins will have the same. Yes. So identical twins will have the same. So if we had an issue where we had identical twins in this classroom, we couldn't tell.
What can tell you?
So not genetics. Their fingerprints will be the same. So you'll have to look for like defects like scars in fingerprints or a palm vein scan, because the deviations in how your veins form in your hand could not always be strictly determined by your genetic makeup, but just by how you're formed in the uterus, right? So things like that can be determined by something other than your genetics. So epigenetic things, is what they would call that, right? Which have you guys been watching on TikTok? If you guys watch on TikTok or if you're watching on Instagram about the effect of basically males, epigenetic, like, so when a sperm comes with an egg, the effects of the epigenetics, so things outside of the genetic aspects of your organism that's being made, or we'll call it a baby for better explanation. But if you're making a baby with somebody, we know genetically 50-50. It has to be, that's just how it works, right? Epigenetically, things that determine how the placenta is formed and things like this for the health of the mother, as well as how that baby's going to be developed, that most of that stuff and the effects of things that would lead to early miscarriages, stillbirths that happen within the first 12 weeks of pregnancy actually come from the epigenetic aspect of the male and not the female. How much have we been blamed for that stuff as women throughout time? Interesting. So all those beers and all those cigarettes and everything that the man might not have been taking care of himself, leading to unhealthy sperm, that all you have to do is pass a test as a man for you to be considered as having healthy sperm is be in the 95th percentile. So we're saying 95% of sperm tested passes the test. Do you really think that it's all actually that healthy and leading to healthy epigenetics as a, okay, that's all I'm gonna say. If you don't know about it, look into it. There's a lot of science looking into that right now that supports that the health of the male actually has more to do with the health of the baby than we originally thought, which is very impressive, and high fives to the ladies out there. Okay. All right. So anyways, so when you cut somebody's DNA, you will get these little fragments, and these are called restriction fragments. So I use this restriction endonuclease to cut at these palindromic sequences. You'll get these restriction fragments as a result. And that the fact that one person to one person is different is called a restriction fragment length polymorphism. And yes, you do need to know that acronym. Be able to recognize it, right? In a multiple choice test.
Not the words, but like what it means.
All right, so restriction fragment length polymorphism is basically describing the fact that if I cut your DNA with a restriction enzyme like EcoR1, that you will have different lengths of product than I will have. So what this means is restriction fragments that are made from cutting with that enzyme are going to vary in length. Polymorphism means different versions. That's what that term will mean. We'll have different lengths of fragments and different numbers of fragments as a result of cutting with the same restriction enzyme from one person to the next. That's what that's talking about. All right. So that's the idea. This is just talking about it in a little more detail. So then we can create DNA fingerprints of what one person's, cuts their products will look like if I cut yours versus if I cut mine versus if I cut the person next to you, et cetera, unless we are identical twins. That everybody should have different fingerprint when we cut them with these enzymes. All right. Way that we determine the size of your DNA. So I keep saying you'd have different size lengths and we can tell the difference. How though? So that brings us to gel electrophoresis. And you guys have seen pictures of these guys. This is not actually a new thing that I'm bringing up to you, but we have seen pictures of this stuff. So basically what this does is because we have so much phosphate, remember our nucleotides, they are sugar, phosphate, and then a base. And we just repeat that over and over again. Because of these phosphates are so negatively charged, DNA has a negative charge to it overall. And so when I put it in a buffer inside of a gel, and I usually make these gels that are a lot like the gels that we grow our bacteria on. It's like semi-soft, right? And it creates basically a webbing, a network of our DNA to travel through. And I put electricity through it. And your DNA, because it's negatively charged, will move to the positive end. Right? Opposites attract. Negatively charged DNA moves to the positive end. But big pieces of DNA get stuck up in that webbing. Just like if I had a big mesh thing and I have all different sides of balls, the big balls won't be able to necessarily pass through as easily as the little balls. Or if you're in a big old mosh pit of people and you're trying to get to the front of the mosh pit to see the band better, little tiny skinny people might be able to squeeze through easier than people might have a little bit more meat on them. All right. So that's the idea. The big pieces of DNA get trapped in that gel and get slowed down a lot more, whereas the tinier bits of DNA are going to move faster through it. So what it ends up looking like is if we put a piece of gel and we put these little wells in it and we put samples in it. And this is Professor Yost. And I mean, I don't know, this is, what do they call John Doe? This is John Doe over here. Okay, whatever. And we run the DNA, put the samples in here, and we turn on our electricity, and the DNA is going to run to the positive end, that the big strands will get stuck up at the top, and the small strands will run out faster. So the closer it is to the bottom, smaller. All right, and you guys have seen these before, yes? Like this is like CSI type stuff. All right, so this is what it would look like. Yeah, so easy enough to imagine that. So if we have DNA from different individuals up here, I want you to look at wells 3 and 4 here. 3 and 4 look very, very, very similar to each other. In fact, you might even say that 3 and 4 might even be the same person or identical twins and something like that. But you can see the difference for the other people from one another. They might all have this big old, band that's here in the middle that everybody has, but there's enough difference between each of these samples that you would be able to tell that they are not the same person. Okay, so that's the idea with that. So how those fragments of the DNA run out after we cut them with those enzymes that cut the palindromic sequences. Those enzymes are called restriction enzymes or restriction endonucleases. All right. So does that make sense to you guys? So if we were worried about this is our evidence, and then, we got evidence from the crime scene that shows that this was blood that was found at the crime scene. Which suspect had the same blood sample? Was it suspect 1 or 2? It's 1, right? And so it's that. Now we can start to see, and the whole reason that happens is that down here at the bottom of this gel, that this bottom has the larger pieces. From after having cut that stuff, okay? All right, do you guys remember how I had mentioned to you guys how in eukaryotic cells, when we make our mRNA from our DNA, we have to cut out this extra junk called introns. right? And that bacteria can't read that. It can't splice it out. So if we put into bacteria this, they can't get rid of our introns. So they can't make the appropriate product. So because our cells will go from our precursor mRNA and cut out our introns, and then splice together our exons to make a readable mRNA that our cells would be able to actually make a product from, right? For translation. We're just get reused by the cell. We can just recycle them. So we can break down the nucleotides and reuse them. So, so it's an interesting question. I don't think anybody's ever asked before, but yeah, fair. So we can take the mRNA out of your cells, not the DNA, but the mRNA that's already been processed and turn it back into single-stranded DNA using our good old friend, reverse transcriptase that we stole from. Do you guys remember where we got it from? HIV. HIV does that. HIV has an RNA genome that it turns into DNA and inserts its DNA into that. So we stole its ability to do that, that reverse transcriptase. We stole its reverse transcriptase and can take RNA now from your cells and turn it into DNA that's already ready to go and be processed and put that into bacteria. And now bacteria don't have to splice out introns. It's already been done for them. It's in the DNA sequence. So we used to have to actually like generate that by hand and like determine what the spliced version was and then make those nucleotides by hand with chemistry, obviously. But now we don't have to do that. Now reverse transcriptase will allow us to take that processed mRNA and turn it into DNA free of introns. We call that cDNA or complementary DNA. It's complementary to what our mRNA is. And we will use this for cloning our bacteria, right? So if I'm telling you about working with all this DNA and making all these copies and putting these things together, a useful tool for all of that engineering, as well as a useful tool for diagnostics, which we'll learn about later in another chapter, is polymerase chain reaction. What's that? PCR. Another biggie that you guys are definitely going to be using, no matter if you're dealing with humans or pets, at some point, a lot of times we'll have to be facing using PCR. PCR is literally just making copies of DNA. That's all it is. It doesn't do anything else. It doesn't make changes. It doesn't actually put it into bacteria. It doesn't do anything else except for making copies of DNA. That's all it does. All right. So this is what I want to remind you of. The whole way this is going to rely on is basically the same exact rules that we have with DNA replication. All right, so it's not just as simple as going from DNA to making DNA copies. Remember how our polymerase worked when we made our DNA copies. We had to add it on to something that already existed, and we could only go in the 5' to 3' direction. So those rules still apply in PCR. We're still using DNA polymerase to drive all of this, okay? This is so sensitive that you can give me one piece of DNA, just one piece of DNA, one virus, and this guy can pick up and detect that virus and make billions of copies within a matter of a couple of hours. But it relies on the same rules as DNA replication. We're going to have to open up the double helix, expose the strands as single strands. So we might have to have it actually spread out as a ladder and then spread it out like this, separating the strands, and then we will use each one as a template. Then we'll have to add primers. This means that once you have separated your strands like this, that if you're trying to make a copy in this direction, you have to add it onto something. And this would be your primer. And we can design the primers to bind to specific sequences. And we usually have our primers be somewhere from like 15 to 30 base pairs, so that they will recognize a specific gene. For example, the primers that are used for detecting COVID will detect the presence of the genes that code for the spike of COVID, and only those genes, not any other gene in your cells, not any other gene in COVID, or genes from flu, or genes from RSV. It specifically targets only binding to find sequences of the COVID spike protein. So whenever I swab your nose and you put it in your solution, and we pop it in the PCR machine that takes about an hour for us to get results, that what it's doing is seeing if the primers can bind, and if they can, then we can duplicate the DNA strand. If the primers cannot bind, you will not get any product. It's literally whether you can duplicate the DNA or not. That's what you're asking. So every time that you put your sample in a PCR machine, you're giving it a primer for a specific thing and asking if it will duplicate the DNA or not. If it doesn't duplicate the DNA, you do not have COVID. If it does duplicate the DNA, you have COVID. It meant the gene was there. Okay, so this is why I try to drive it home to you guys. You don't just take a swab of somebody and put it in a tube and plug it in and play it, say, test for all of these things. You have to have the individual primers that are there. And if you have a whole bunch of primers in there, like if you're putting in together flu primer, COVID primer, RSV primer, pertussis primer, all of these things, all it's going to tell you is that, yes, you have something of one of those things, but it can't tell you which one. So you have to have it designed to be able to tell you which one it is as well. And so it gets, you can only test for one thing at a time, basically is what I'm telling you. All right. So you have to have primers for it as well. So if somebody comes into your clinic and they have a more exotic disease, but your PCR machine, you don't have the primers to test for that exotic disease, guess what? You ain't doing PCR to test for it, or you're sending it somewhere else to do the PCR testing, but you're probably going to have to figure out other ways to test for it. And that's where all those other tests that we learned about in the lab come into play. And like I think I've already mentioned, a lot of times when we're running these tests for the lab, we are confirming everything with PCR, like they're cross-confirming. You don't just go based on one test, right? So this is something that we'll do. All right, so we've mentioned our primers, our DNA polymerase. What we're gonna be doing is basically using heat to separate our strands every time. So we'll use heat to separate these strands out. So then we can attach our primers onto whatever sequence they match up to and build off of it. And that's pretty much how this whole thing is going to be working. But because we're exposing this to very high heat temperatures, we're talking about 92 degrees Celsius. So it's almost boiling temperatures. Most DNA polymerases can't actually survive. That's too hot for the protein to withstand. So we use DNA polymerases from bacteria that we got from like those deep thermal vents that are like super hot above boiling temperatures. If they can survive that, they must be able to replicate themselves. If they can replicate themselves, they're replicating their DNA, which means their DNA can withstand those temperatures. So we've stolen from those bacteria, and now we use their DNA polymerase in this. We call it Taq polymerase from Thermus aquaticus. So this is how this works. We take your DNA sequence, we heat it up. This step here, separating the strands, is called denaturation. All right, then we'll cool it down slightly to cause our primers to anneal. So this what this step is showing here. Our little golden guys are our primers. And again, they're going to recognize specific sequences for specific genes. They're designed to be very specific. This step is the priming step or the annealing step. On the exam, I'll usually call it anneal. Anneal is bringing things together, causing them to stick together, okay? So the annealing step involves the primers binding. Once our primers have bound, which is this step right here, the next thing that we can do is extend. So that extension step is DNA polymerase adding the complementary nucleotides. All right, so we will add our complementary nucleotides until we get our product. And then what do? Heat it up and do it again. We'll heat it up and separate our products and then make copies of those. And we'll keep doing that until we have enough that we think it will be at a detectable level. Making one copy, so going from one DNA strand, double-stranded DNA, to two is not enough for us to be able to run on a gel and be able to visualize it, or for our purposes to say, yes, this is actually making appropriate copies of this. So we want to be sure that that's reproducible and that if you're using this for genetic engineering purposes, you're going to need a lot of DNA to work with. So we can take that one copy and turn it into billions by copying that. Each cycle doubles. So if you had one DNA molecule, let's see, if you had one DNA molecule that gets spread out, open up the two sides of it, you make a copy of that to leads to two. So one becomes two, this guy becomes two, and this guy becomes two in the next cycle. So it's four. What do you think the next cycle is going to look like? 8. Yeah. So how many would you have after 4 cycles? 16. How many would you have after 5? 32. How many after 6? 64. Okay, we starting to see that pattern there? You better believe that on the lab test, I'm going to ask you to do that calculation. I'm going to say, if you start with 1, how many strands would you have after this many cycles? I'm not going to make it 452 cycles. I'm going to make it like 5. Okay. So you should be able to just do that math on your own if you didn't memorize this. Okay. Should be basic. I guess there's another math thing I didn't warn you about. Again, I'm not going to play this video because I don't feel like we have the time. But if you guys want more clarification on how PCR works, the videos are really good at providing clarification on those things. Okay. So basically, we can use all of these tools, cutting at specific sequences, making these random fragments, or looking for specific genes with our PCR, or amplifying specific things to make sure we have enough copies of something, and then cutting the copies that we made, and then putting them into plasmids or things like that we can then put into our bacteria, and now shuttling our genes into other organisms. This is recombinant DNA technology, removing genetic material from one organism and combining it with a different organism. The things that we make as a result are called clones. Cloning involves removing it from the genetic donor, putting it into a vector of some kind, it's usually a plasmid or a virus, and then that plasmid or virus will carry that gene into the cloning host, usually bacteria or yeast, which will make the product. That's cloning. All right, so we already talked a little bit about restriction endonucleases. We talked about making sure that our DNA doesn't have introns in it. So that's our complementary DNA from using reverse transcriptase. And we talked about amplifying using PCR. Plasmids are circular bits, extra bits of DNA that have extra genes on it that usually help for bacterial survival, but we're manipulating that system and turning in whatever gene we want into it and forcing bacteria to use it. Bacteriophages can also be used through that idea of that lysogenic conversion. And when we are using that in genetic engineering, we're going to call that transduction, basically using the bacteriophage to introduce new DNA to our bacteria that way. We use plasmids or bacteriophages to do this. This is an example of a plasmid. Here we have a segment where we would break up a gene. Each of these represents a gene, or this is the area where we have the origin of replication and the direction that we would be doing that in. But basically, this is an ampicillin resistance gene, and this would be a gene for something else that we will split up that gene. And if we are able to insert new DNA in here, it'll break up that gene and that gene doesn't work anymore. And that's how we know we inserted the DNA in there. Okay, that gene won't work anymore. This gene over here, this will tell us that our bacteria actually picked up the plasmid whenever we introduced it to it. So that plasmid got introduced, now your bacteria is ampicillin resistant. So we can treat your media with ampicillin, and only the bacteria that took this plasmid into them will actually grow in the media. So now you're only growing bacteria that took the plasmid, and if that other gene doesn't work anymore, then we would know that means the plasmid also took up your DNA that you put into it. So now you're double checking to make sure that stuff works. We'll go over that in more detail in lab 10. Okay, again, if you are taking a plasmid and you're taking a bit of DNA and you're combining them, essentially cutting them with those restriction enzymes and putting those things together, they don't automatically seal these bonds together. What are we gonna have to do to seal the bonds together? The same thing as whenever we're making our lagging strand, the DNA ligase. It seals those sticky ends together so it actually operates the way that it should. Remember, reverse transcriptase is basically taking our processed mRNA and turning it back into a DNA version that we can use in our bacteria. This has the introns removed. All right, so you take your gene and you'll take your plasmid. All right, so once you have identified what gene you want to put in the plasmid, now we have to have enough copies of each of those, which you might make by your PCR. Then we're going to cut each of those to make the appropriate sticky ends. And then we'll cut our plasmid as well. And then when we cut our plasmid with that sticky ends, we'll be able to stick our DNA into that plasmid. and force that recombinant plasmid into the target cell. This is a process called transformation. That's whenever we take up outside DNA. All right, and then that cell will just grow it up and it'll make from that gene, it'll transcribe it into mRNA, it'll translate it into the protein product, and then we will, in our flask of media, collect that product and be able to purify just that product and then give you your insulin shots that you made from bacteria expressing human insulin. Okay. That was one of the first ones that we had. We've also already talked about basically making these cuts in different sequences and looking at the different fingerprints that we can get as a result, telling the difference between people. But we can even use our understanding of nucleotide sequences and then basically make random primers and then see what we can build off of those primers as a result and then sequence those things. We can also use our primers like probes. For example, if we had the same primer that was 30 nucleotides that only bonded onto a sequence of COVID a genome, then that could also be used to take somebody's cell samples and see if that primer just bound in there. Not that it even had to multiply, but did it bind where it was supposed to? And so we can add onto the primer a fluorescent marker and then treat the cells to see if that fluorescently bound marker binds with COVID primer on the cells to see. That's called nucleic acid hybridization. There's a picture that'll explain it better than I'm explaining it. Hopefully it has a picture of it on here. But basically you have a sequence of DNA that might look like this, and I have all your cells, and I will treat them in a way to open up your cells, stick this in there, and if it stays stuck, it means you have this sequence in your DNA. It bound complementary to it somewhere. So I can see if you have the gene or not in your DNA, or in the bacteria's DNA, if they have the gene or not, or whatever. So it's just seeing if it will have complementary binding to a known sequence. This is essentially gene probes. And so we can link them to enzymes that'll lead to color changes in solutions, or we can link them to fluorescent markers that literally glow a color, things like that. Okay, so we can use this to diagnose, to see, hey, I got a specimen from my patient. Do they have genes from E. coli in their sample? And I can just see if it hybridizes or not with a DNA sequence. I can also do that to see if you have a genetic defect. Do you hybridize with this specific genetic defect? This is actually a really great way that we can use to look at different kinds of cancers. So different kinds of cancers might be associated with different kinds of mutations. Does this mutated probe bind with your patient sample? If it does, they have the mutated version of the gene. Okay, so it's gene probes. How good this works. So you have your sample of your cells. You'll expose them to this DNA that's been labeled with fluorescence or something. If it binds onto the cells, then that cell has that particular gene. Okay. If it is involved in fluorescence, and you can do this in an actual tissue sample that you have taken from your patient, like you have taken a biopsy from your patient's breast cancer, and you have put it onto this plate, you've washed your cells and you've prepared them, and now you're going to put this probe on there that is literally made to glow fluorescent green. And if it sticks to your cells, your patient has a mutation. Now we can do that in your actual cell sample from that biopsy. That is called FISH, or fluorescent in situ hybridization. We're using actual cells from that living patient. We didn't have to kill the patient to get the cells. So we call that in situ. You guys know what in vivo means? It means in the living organism. So actually in that organism. So when you guys go get your skin tests for your tuberculosis, that's a test that we can do in vivo. We're doing it in your living body and seeing your living response to it. So it's in vivo. We have ex vivo, which is outside of the body. In situ, it's using it from the living organism, and all these terms that can be used in that sort of thing. But as long as you know that FISH stands for fluorescent in situ hybridization, and we have matching of DNA sequence, and that leading to the resulting aspect of whether you have the sequence or not, basically. And we can put all these different versions of that on a big old plate. So this would be a plate that has like 364 different versions of mutations for different cancers or for different diseases or whatever. And then I can put your sample, mix it up with the plate, and whichever ones that it binds to, you're positive for. So we can do a whole mess of testing for all different versions of disease on this one plate. Extremely expensive, but it gives you an idea of what type of mutation might be causing your patient's cancer, or which version of this disease you might have, all from one test. All right. Cutting up somebody's DNA, getting those different size bands. This is the idea of creating a DNA fingerprint for those people, just coming back to that. And we can create sequence maps of whole organisms basically by using random primers throughout the genome and looking to see what happens if the nucleotides can build off of them and which ones are building in what order. And then that's how we can compare genomes. So human genomes share approximately 80% of our DNA with mice, 60% with rice, and 30% with C. elegans, which is a very commonly studied worm in genetics.
Wait, I'm confused. You told us we're more related to mice than more like other animals?
Depends on the animal, right? So I don't know what our exact relationship is with other things, but like we know that with chimpanzees, we share like 99% of our genetic code with chimpanzees. And we can already see the difference there, right? So that is really puts it in a perspective. All right. And we'll use all of that basically to try to understand what each gene might code for and what the differences in each gene can mean for each person. So there's a gene for eye color, and there's all different versions of that gene that you can have, for example. So this is trying to study the differences that are possible within all of the different genes, understanding what each of your genes code for. And again, any mutations that can lead to variabilities that could lead to problems. That's genomics, basically, studying those genes. And we talked about already that if you went from AAA to AUA, you are coding for a different amino acid as a result. And therefore, your protein product would be different. And now this could be detrimental to an organism, especially if you have changed from one amino acid that has this big old R group that is entirely hydrophobic, and now suddenly you've replaced it with something that has a big old fat acid charge to it. And if that happens, you're screwing up how your ribbon folds whenever we're talking about our different levels of protein structure. So one nucleotide change, just one nucleotide change can be deadly. One nucleotide change in your entire 3 billion gene base pairs, one nucleotide change in just all of those billions of base pairs can lead to something that could be potentially deadly. An example, and that's called a single nucleotide polymorphism. Again, polymorphism means that there's some sort of change that happened. So if it's a single nucleotide change and the effect of that change, all right, so these can be something that we've already talked about with our different kinds of mutations. You can have a single nucleotide polymorphism that leads to a different amino acid, but maybe your protein still folds just fine and works just fine. Maybe you have a change in your amino acid that doesn't change your actual, you're changing your nucleotide sequence that doesn't change your amino acid, and there's no change at all. Who cares, right? That's silent, right? But if you're changing it to something that alters the actual structure and potentially function of that protein, that's where we'll get genetic variability from, but it's also where you could potentially have deadly mutations as well. So our example down here is that in thrombophilia, we have an issue with clotting, where you have a massive increase in clotting in your blood due to one nucleotide change in a genome. One nucleotide change mutation in that person's DNA has led to them now having a clotting disease that they can likely have a very highly increased likelihood of having a stroke and dying. All right. So this is explaining that to us, that if your DNA said GCT, the complement to that, CGA, coding for arginine, that's normal. But if we had, instead of GCT, GTT, now suddenly we're coding for glutamine, and that instead of having arginine there and having glutamine instead, we can't have proper control of clotting anymore. We have too much. All right. Again, so we introduced that concept of microarrays, which show those plates with those little dots of the different kinds of single nucleotide polymorphisms or different mutations that could represent different kinds of cancers or whatever. And I can take your patient sample, mix it with that microarray, and basically weed out which kind of cancer your patient could have based on their genetics without having to sequence their whole genome. Just seeing if they match up these already known mutations for these things. All right. So we can use this to create products that if we already know the sequence for, we can have bacteria make it for them. We have bacteria that will make interferons for us, which are things that we would use to That's our signaling molecule. Remember, we talked about viruses. We can send a signal if you have a viral infection to the guy next to you. Hey, Eddie, we got somebody breaking into our cell here. Get your shotgun out and be ready for the invaders that are coming. That's an interferon. So we can actually have bacteria make this, and then I can give you shots of interferon that just gets your body more primed and more likely to fight off viral infections, and all sorts of things. Things like remicated Humira. These are biologics that are made cloned like this, and then we can have other organisms make that product for you, and their antibodies that we can make bacteria produce them. And now we can give you cost-effective infusions of these antibodies that we didn't have to get from like animals necessarily or whatever. So we'll talk more about that stuff later though. And we use this stuff to make vaccines and other things like that. So this is just to finish up kind of this concept. A GMO is any organism that has been genetically modified for whatever purpose, either to make it more resistant to pests that could be attacking them, more resistant to disease, and all of these things that we might see in our foods, right? Foreign genes will be inserted into other organisms. This creates genetically modified bacteria that make products we want, plants that are more resistant to pests, or animals that might be modified for whatever reason. An example of this is that we often have corn that has genes from Bacillus thuringiensis that makes them less susceptible to insects causing issues. So we don't have to treat now with pesticides as much. Okay, but when you eat this organism, when you eat that corn, and I'm going to tell you, most of the foods, especially plant-based foods that you're going to be eating here in America, are GMOs. Probably in most parts of the world, you're going to be eating GMOs. It's hard not to eat GMOs nowadays. Okay, but these organisms have these genes inserted in them. You're not going to get those Bacillus thuringiensis genes incorporated into your genome. That is not the risk. When you eat an organism, you eat it, you digest it, you break down their DNA. It doesn't matter where that thing came from. You're breaking down its DNA, and you're just going to use its components to build your DNA. That's what you're doing with that DNA. There's no risk of it jumping into your genes, right? Doesn't matter where it came from. You're just going to break it down. Your body's made for that. So you're not going to incorporate those genes into your genome. The risk here is that if I put this gene into the corn, that plants have a mechanism in them called transposons that can allow them to share their genes with other plants that are nearby. And now suddenly you have wild plants that are now resistant to these pests that depended on that wild plant for food, and you've disrupted an entire ecosystem. That's the problem with GMOs. There are problems with GMOs. You're not getting those genes in your body. That's impossible. Don't let you or your mom or your dad go on talking about how you're going to get these weird foreign genes inserted in your body by eating a GMO. That's not the problem. The problem is this stuff, disrupting ecosystems, okay? So take that idea home with you. There's a video on GMOs if you want to learn more about them. We can also, this is the last bit of it, basically take that we know that you're supposed to be coding for insulin this way, and now use all of this knowledge that we know about DNA and transcription and translation and create working versions of these genes and try to reintroduce it into your body so that your body can make working versions of these proteins that you are not able to make because you have a genetic defect to it, okay? And the idea is that this will be permanent. That's the goal with these. Now we do have things where we are going to introduce something like HIV. We already know HIV can insert its genes into your genes. Imagine if we could hijack that mechanism and cause HIV to insert working genes where your genes don't work. And now your cells can make a working product. This is real and happens in real life, and we are saving lives this way currently. There are different versions of disease where we can take a normal gene from a healthy person, clone it into a virus like HIV, take the sick person's bone marrow, mix that virus with the good gene. No HIV stuff is in it anymore. We hollow it out. We put the good working gene into HIV, mix it with the sick person's bone marrow. We'll let it infect their bone marrow and then infuse them back with their own bone marrow with the working gene that's been incorporated into its DNA. And that works and can permanently alter that person's DNA. This does not spread, though, to their sex cells. So it's not heritable. So we can fix them and their cells of their actual body, their somatic cells, but we can't fix your gametes. So somatic gene therapy is a thing that we have right now where we can change the individual, but it is not heritable. Germline therapy is therapy where we would have to actually do that genetic manipulation on the egg or the sperm or an early embryo. This is something that we could do in fertilized stuff through IVF, that before we implant that embryo. if we saw that there was a genetic defect in there, we could use some of this technology to repair the genetic defect. That comes back to that conversation we had at the beginning of class, though, of where do we draw the line of the ethics of changing up people's genetic stuff. You want your kid to be, a basketball player, so you've given them basketball player genes, and you can afford to do that for your kid. But what about the people that can't afford that? Is that right? Is that fair? And where do we draw the lines of that stuff, right? But obviously, if somebody has one of these diseases like we have, acute lymphoblastic anemia is what this was, or inherited vision loss, or spinal muscular atrophy. And these diseases are, as of 2022, which is years ago now, able to be effectively treated through gene therapy. We can cure you of your mutation, basically, and make you basically your body function in those aspects normally. It is not heritable though. So we would have to do any heritable mutation changes in the actual early embryo before it gets implanted in the uterus.